Evidence for semi-conservative replication. Explain why the process of DNA replication is called semi-conservative. Interpret data from Michelson and Stahl experiments. Okay, Watson and Crick worked out the structure of DNA. They realised the semi-conservative replication, but they needed to prove it because there were two possible methods. Conservative, where the parental DNA remained intact. There was a separate daughter DNA copy built from new bases. Of the two molecules made, one was a new material and one was old material. Semi-conservative is where the DNA is split into two separate strands, and each replicated its mirror image or missing half. So both the two new molecules have one strand of original material and one strand of new material. Now we know it is in fact semi-conservative replication. So the experiments that Michelson and Stahl did, they knew that there was a distribution of old DNA that was different in each model to find out which one was correct. So they looked at the distribution of the old DNA and the new DNA and found out which type of replication was correct. They labelled the old DNA and look how it was distributed after replication. So they worked on three facts: that all bases in DNA contain nitrogen. Nitrogen has two forms. It has the lighter nitrogen-14 and the heavier nitrogen-15 isotope. And they know that bacteria incorporate nitrogen from the growing medium into the new DNA they make. So first of all, the bacteria is originally all made of heavy nitrogen-15. But then, if they transfer it onto nitrogen-14, which has lighter DNA, you can see the difference. So they labelled the old DNA, the original DNA of the bacteria, by growing them on nitrogen-15, the heavier DNA. They then transferred the bacteria into nitrogen-14 for one generation to allow for one replication. And then the mass of each new DNA molecule depends on which method of replication. To separate DNA types, they centrifuged and extracted the DNA. The lighter the DNA would be nearer the top of the centrifuge tube, whereas the heavier DNA would be nearer the bottom of the centrifuge tube. And they also analysed the DNA after 2 and 3 generations. And here was the results. So, the original heavy DNA was nitrogen-15. So that was what the original strands were. And then they found that the first generation was all made of half and half. All of it went to the middle. The second generation, half of it went to the middle and half of it went to the top, because half of it was completely light, because they can only make new strands out of nitrogen-14 now. And then in the third generation, they found that there was less of a percentage of the middle band and more of a percentage of the lighter band. So they found that in the second generation it was half and half, but the third generation it was almost 75% light DNA and only 25% of the half and half.